Should we wait? Yeah. Okay. Well, good afternoon, everybody. Thank you and welcome to our seminar series. It's my pleasure to introduce today's speaker. Although we have never met in person, we have read each other's papers for decades. Professor Robert Thorne from Cornell University, a dear colleague and that has done tremendously important work in the same area that we have worked in, basically what I call controlling the fate of water, and which phase it goes to determines what comes out at the other end of the biological system. And his interests are two-pronged. He is an expert in the physics of ice nucleation and growth, that he uses that expertise to eliminate it, to understand better ways of doing crystallography and understand biological structures and functions. But he also uses the same principles to eliminate ice or control it so that we can have cryobiology or preservation of living systems. And today he's going to share with us some of his recent work in eliminating ice for cryopreservation of oocytes and embryos, which is a fundamental aspect of, as you know, in vitro fertilization, a field that needs tremendous need in automation and better ways of doing processes. Without further ado, I'll pass it to you, Robert, and let's hear from you. Thank you for joining us.
Well, thank you so much for the invitation. I haven't been able to go to many conferences for quite a few years now, and so I haven't met most of you, and I'm really happy to have this opportunity to talk to you and to learn from you. The work I'm going to be telling you about has occurred under different, with different ends in mind over the last 20 years or so, but the more recent work that focuses on oocytes and embryos has been done in collaboration with Soonhan Chong, who's head of theriogenology in our vet school at Cornell. So I'm going to focus today on, first by talking about cryopreservation of small things and what are the current approaches. And by small, I mean things that have a minimum dimension that's somewhere below 1 millimeter. And that means things like oocytes and embryos and maybe, thin tissue samples or cell suspensions, but definitely not organs or anything remotely big. So I'm going to talk about key ideas relevant to ice formation in the regimes of cooling and warming that you can achieve when you have very small samples. So what happens when you cool very fast and warm very fast? That's quite different than what happens when you cool at more gradual rates. Then we're interested in doing something about cryopreservation of living things. And it turns out that a lot of the problems that you folks have been dealing with are similar to problems that have been dealt with quite a while ago in the context of X-ray cryocrystallography. So I'll talk about some tools and methods from there. And then we're going to apply those methods and use X-ray diffraction to study ice formation in bovine oocytes and embryos. And the key takeaway is that you can do ice-free cryopreservation, so completely ice-free cryopreservation of small permeable samples. And so now we're looking for collaborators to see how far this can go. So in cryopreservation of small things like oocytes and embryos, there's been two approaches. The older approach is a slow cooling approach. You typically use some CPA, some cryoprotectants, but not too much. And you cool very, very slowly, at least initially. And the idea is not to eliminate ice formation, but rather to have ice form in the extracellular spaces and have that ice grow so that it sucks water out of the cells, dehydrating them until the concentration of remaining water is so low that the intracellular water can be vitrified. And this works remarkably well for things like embryos, but when you get to single cells, failure rates are often too high. So the approach that has revolutionized human-assisted reproduction is the vitrification or rapid cooling approach. There you use much larger cryoprotectant concentrations and large, but not ridiculously large cooling rates but they're still a few orders of magnitude larger than in the slow cooling approaches. And the goal here is to cool your sample without forming any extra or intracellular ice. So all the water in your sample should vitrify. And the success rates can be quite high, especially for human and mice oocytes. Other species methods are still in development, and it remains to be seen. So this vitrification approach, you're trying to avoid ice formation. And a reason for that is that it's generally thought that formation of ice is a major cause, if not the most important cause, of damage that leads to poor post-thaw outcomes. So how are cells damaged by cooling and thawing? So when ice crystals nucleate and grow, those crystals can do mechanical damage as they grow, disrupting cell membranes, the cytoskeleton, spindle apparatus. Another key thing is a chemical, and that is results because ice crystals are essentially pure water, and as they grow, they reject essentially all solutes from cytosol. And so those solutes become concentrated in the remaining uncrystallized solution. And that includes salts, proteins, I mean, anything, everything but water will get concentrated. And as you increase all those concentrations, pHs, water activity, lots of parameters that affect, for example, protein conformation and folding may be changing. And so you can get precipitation, aggregation, and unfolding in response to this, as well as changes in structure and function, perhaps, of biomolecular complexes within the cell. And some of these changes may be irreversible, meaning when you thaw, they don't come back. So that's all from effects of cooling and warming on via ice formation. But even if you don't have ice formation, cooling and warming could, in principle, still cause problems because proteins have a stable form only in a range of temperature near biological temperature, and at lower temperature, they can unfold. They typically will try to unfold. Membranes may undergo phase separation. There's a lot of things that can, in principle, occur to the molecules and assemblies of molecules just as a result of varying the temperature. And this is just showing you have a protein in its native form at room temperature. And then as you get down somewhat below the freezing point of water, they'll typically unfold. And then those unfolded proteins or partially unfolded proteins can form aggregates that can be irreversible. And another problem that occurs, more of a problem when you're cooling big things, but can still occur in small ones, is that you can have microfracturing or cracking due to differential contraction of the sample's components and also of the sample relative to the support it's on during cooling and warming. So the simplest idea to optimize cryopreservation of small things is to cool and warm so fast that no ice forms ever, so that all of these things that can go wrong just because temperature is changing don't have time to go wrong. You basically outrun them because you're cooling and warming so fast. And at the same time, you got to avoid sample fracturing. So that's a very simple idea. So how fast must you cool and warm to avoid ice formation? Um, well, the first answer is infinitely fast. If you look at the homogeneous nucleation rates below the homogeneous nucleation temperature for ice, they're absolutely enormous. And even in a human oocyte, the number of nuclei that will form at any reasonable cooling rate to cryogenic temperature is going to be huge. And so you're always going to have at least lots of nuclei inside. You can never avoid that. But fortunately, ice nuclei are really tiny. They have about 100 water molecules in them. They're about a nanometer in size. So it's okay if they form as long as you don't give them a chance to grow. Okay, and we'll see in a moment that their growth rate drops as temperature drops. So another answer is to this question of how fast you have to cool and warm is that it should be fast enough so that the ice fraction is not detectable. And in practice, that fractions is below about 1% or so. Depends on your measurement method, but that's a good rule of thumb. And that gives an operational definition for critical cooling and critical warming rates. How fast you have to cool or warm so that the ice fraction isn't detectable at some threshold. So the critical cooling rate for pure water is enormous. It's about 1.5 times 10 to the 7th degrees C per minute. You can't achieve that except in sub-micron thick films. So that's kind of out of the question. And the critical warming rate for pure water, I don't know what it is. I don't even know of an estimate of it. It's going to be huge. So then in terms of these potentially irreversible biomolecular and cellular changes that might occur during cooling and warming, their time scales are probably range from the tens to hundreds of microseconds to, for larger changes, much longer than a second. So we can do something about these enormous cooling and warming rates that we need for pure water by adding cryoprotectants to our samples. If you use penetrating cryoprotectants like ethylene glycol or DMSO, they go inside the cell and interact with water inside and do so in a way that reduces cooling and warming rates required to get no ice. Non-penetrating cryoprotectants, as you know, things like sucrose, dehydrate the cell and thereby increase the concentration of everything else inside the cell. And that includes whatever penetrating cryoprotectants you have, as well as whatever solutes are inside the cell, all the proteins, carbohydrates, everything but water inside the cell. And it turns out all solutes have cryoprotective properties. So you get some help relative to pure water, not just from your cryoprotectants, but from everything else that's inside the cell. And the way cryoprotection works roughly is that around any solute in water, the water will interact with that solute and the structure of the water will be perturbed by that interaction away from the structure that's favorable for ice crystal formation. And the water may also form hydrogen bonds, which is often the case with cryoprotectants. And it turns out lots of studies have shown, including this paper from my group, that bound water present in the first and perhaps second hydration shells around solutes doesn't crystallize. So basically, you don't have to worry about that bound water. All the problems are going to come from the unbound water in the cell. And so the relevant parameter governing ice formation is something called the free water fraction, which is the fraction of sample volume occupied by unbound water. Okay, so the critical cooling rate for pure water is huge. As you add cryoprotectants, the critical cooling rate drops. And measurements of critical cooling rates suggest that the critical cooling rate varies exponentially with the concentration of whatever solute or cryoprotectant you're using. There's a simple way of a simple analysis that leads to that conclusion, or leads to that formula, that dependence. First, we're in the rapid cooling. If you're at the critical cooling rate, you're cooling very fast. And in that fast cooling, you're almost certainly going to be nucleation limited and almost certainly going to be homogeneous nucleation limited. And then if you do a little stat mech, you can convince yourself that solutes will reduce the probability of forming a critical nucleus, and that very simply gives you this exponential dependence on concentration. What is that exponential? What's the practical implication of that exponential dependence? So we know some CPAs like DMSO are toxic, or at least they're thought to be toxic at high concentration. So we'd all like to reduce that. Oh, I got to move a little bit here to get the lights to come back on. That's okay. Guess I don't have to have them on. So if you want to reduce the CPA concentration by a factor of 2, it turns out you're going to have to increase the cooling rate by 2 orders of magnitude. Okay, so it's a pretty steep variation in critical cooling rate with concentration. So if you want to do much better than we currently do, you really got to jack up the cooling rates. So what about critical warming rates? They're generally much larger than critical cooling rates, anywhere from 1 to 4 orders of magnitude and perhaps more. We don't actually have a lot of great data measuring these because measuring that data is pretty hard. But they're much bigger. And that means that it's going to be way harder to avoid ice formation when you're warming something than it is when you're cooling it. And in fact, evidence dating back to Peter Mazur's work and probably earlier, lots of evidence suggests that much or most damage in cryopreservation of small things happens during warming and not cooling. So why is this the case? Why are critical warming rates so big? So when you're cooling, when you're cooling fast, generally the first thing that happens is ice nucleation. And if you're cooling fast enough, not much happens until you get near the homogeneous nucleation temperature, around 235 K. So then ice crystals form, and then those crystals can start to grow as you cool further. And it turns out that the nucleation rate, by the time you have lots of nuclei, the growth rate is falling. And so those nuclei may not grow that much. So you end up with a cold sample that might have a lot of nuclei, but the volume fraction of ice may be quite small because all those nuclei haven't grown much. On the other hand, during warming, all of those nuclei are available to grow as soon as the solvent, the water molecules develop mobility, which starts above the glass transition and really takes off above about 180 Kelvin. And so those pre-existing ice crystals can grow like gangbusters during the warming phase. You could also, in principle, have additional nucleation. If there's enough time, larger crystals will tend to grow at the expense of smaller ones. And eventually on warming, you know that all of that ice that forms is going to melt. So the ice during warming is transient. That makes it a lot harder to see when you're doing in cryopreservation practice. So the bottom line here is that at typical cooling and warming rates that are used today, the maximum ice fraction is going to be a lot warmer during warming than what forms during cooling. And if you want to do something about that, you have to warm a lot faster uh, uh, than we currently do. Now, that's not the whole story on critical warming rates. The other really important point is that the warming rate you need to keep the ice in your sample small depends on how fast you cooled the sample initially. And this is an experimental fact that we've verified since this paper that was published over a decade ago. And it's probably connected to the fact that the faster you cool, the fewer nuclei you have. And therefore, when you warm, there are fewer nuclei, fewer crystals that are growing. And therefore, if they're all growing at the same rate, roughly, you're going to have a smaller ice fraction after a given warming time. So this gives you another way, not just cranking up the warming rates, but actually if you crank up the cooling rate, that'll help pull down the warming rate you need to not get ice. So to summarize this background section, to minimize ice formation and related cellular damage in cryopreservation of small things, you want to cool as fast as possible, warm as fast as possible, and use enough CPA, which means not too little that the sample fractures or too much that CPAs kill it. All right, well, I don't think you needed to listen to what I've said so far to know that that's true, that that's a good thing to do. But hopefully I've given you a little more motivation for it, or clearer motivation for it. And so let's think now, well, how do you cool as fast as possible? Basic principles are obvious to engineers, at least. You minimize the total thermal mass of the sample and of whatever's holding it and of whatever liquids around the sample. You want to maximize contact of the cooling fluid with the sample, although actually there's some complications because of Leidenfrost effects, which actually someone at Minnesota published in crystallography on that problem. And then you want to maximize the convective velocity of the cooling fluid relative to the sample while you're cooling. And then this is something that certainly wasn't appreciated in crystallography, cryocrystallography, and it's still not appreciated in cryo-electron microscopy, is that when samples are small enough, they will cool in whatever cold gas is present above your liquid nitrogen or your liquid ethane. And in the case of nitrogen, there's a lot of cold gas, invariably, at least a centimeter and often several centimeters of cold gas. And once your samples get below about 1 millimeter inside, actually most of the cooling can happen in that cold gas before the sample ever gets to the liquid nitrogen. And so it's really important when you're dealing with these smaller samples that you get rid of that pre-cooling. Okay, so that's how you cool as fast as possible. How do you warm as fast as possible? Well, it's all the same principles. Minimize thermal mass, maximize contact of warming fluid with the sample, maximize convective velocity, and eliminate pre-warming in the air before you insert it in the warming fluid. Okay, so there's nothing earth-shaking here. And as physicists, theoretical physicists like to say, the rest is engineering. Of course, for us, that's the fun part. And yeah, that's what we've spent a lot of time on that. So how much better can we do in cooling and warming over current practice in assisted reproduction? Well, if you look at the samples that the two fields deal with, they're about the same size. There's complete overlap in sizes between oocytes and embryos and the sizes of crystals used in crystallography. And there's also overlap in the water contents. Average protein crystals have 55% water content by volume. And the record, I think, is actually 98% water that someone got a structure from. But in any case, 70% is not uncommon. And then the basic steps: harvesting your samples, serial soaking them in cryoprotectants, putting them on a support, plunging them in liquid nitrogen, and then storing them. It's all the same stuff. But since in crystallography, we do enormously more samples than are done in assisted reproduction, there's been a push to develop good tools. And so on the left is shown are the sample supports we use that take the place of things like cryotops and straws. They consist of a microfabricated thin film. It's about 10 microns thick. There's no plastic except for where the sample goes. Everything is nicely thermally isolated. It's exactly sized to match the sample. So you get very little surrounding liquid and excellent contact with the cooling fluid. And these aren't new. In fact, my group that invented them, first prototypes in 2003, published in 2004. That's when I started my company. And our first products were making these little sample supports. And then in addition to that, there's a whole suite of standard tools in which those supports are inserted and then stored in and shipped in. So there's a whole supply chain that's been worked out for cryocrystallography, or whole handling chain for cryocrystallography. And if you look at the image on the right, you can just see that cryotops are enormously massive and not optimized in any way for maximizing cooling or warming rates. And that's true of all the current commercial products. So then how do you cool really fast? Okay, well, maximize convective velocities, get rid of cold gas layers. So we got, many years ago, we got an NIH SBIR grant to develop this instrument for crystallography. It holds, you put 4 pucks in here, that hold 64 samples. And I'll just show you a little video of what it does in action. And this video was made in 2015, which is how long we've been selling these things. So the sample's on that wand that he's retrieved the sample from under the microscope. He just sticks it in there, loads it, and then the sample's automatically plunge cooled, cold gas is removed, all that stuff. And then it sample's automatically loaded into a carousel. This is way more than is necessary for what we're interested in here, but it is necessary in crystallography. So how good does this work? If you do hand plunging in a typical liquid nitrogen dewar, foam dewar, and you're doing, doesn't really matter how small the sample is because your cold gasket layer limited, you might get cooling rates of about 30,000 degrees C per minute, which coincidentally are about the cooling rates that people have reported for oocytes and embryos on cryotops. So with this instrument, cooling rates are a few million per minute. So about a factor of 100 improvement. Of course, you could do much better hand plunging if you know exactly what you're doing, but most people don't. And actually doing it reproducibly is pretty hard. You really need good hardware. And as a measure of just how well this works, we've shown that using this instrument that we can vitrify pure water for use in cryo-EM work, which is something that Nobel Prize winners said didn't seem to be possible. Okay, so now with these tools from crystallography and these insights, we decided to look at ice formation in bovine oocytes and embryos. They're about the same size as human oocytes and embryos, the lipid content's higher, and lipid content is thought to correlate with being harder to preserve. And they generally have significantly less success with bovine, but that could just be, variations in protocols. Hard to know. So what's our experimental protocol? So we cryoprotect these by soaking in equilibration and vitrification solutions. What I'll call 100% strength solution is the standard full strength vitrification solution, which has 15% ethylene glycol, 15% DMSO. And then we dilute that down to 40% of full strength. And so we try a range of cryoprotectant concentrations. And then all of the solutions will have sucrose in them as a non-penetrating cryoprotectant. Then we'll mount these either on a cryotop, or there should be an ore in there, or on a crystallography support like the one I showed you. And then if they're on cryotops, we'll plunge cool them manually. And if they're on crystallography supports, we'll plunge cool them using that automated instrument. So this will give us two groups for further discussion, what I'll call a standard cool group using standard tools and protocols, and a fast cool group. To warm these, we wanted to look what was going on in the X-ray beam, and so it wasn't easy to just plunge these into a bucket of water. So we, in fact, warm them in the X-ray beam using a room temperature nitrogen gas stream. And that's how we're going to warm. And then we're going to shoot X-rays at these oocytes and embryos after they've cooled and before we warm them, to see how much ice formed and what the character of the ice is that formed during cooling. And then we're going to warm them up and warm them fast, not slow. We're going to warm them fast and use time-resolved X-ray diffraction to observe the evolution of ice during warming. And these experiments, by the way, directly parallel experiments that we did many years ago using protein crystals, trying to understand ice in protein crystals. We did our experiments. It's at the Cornell High Energy Synchron Source. It's a ring right here on campus, about a 10-minute walk from the physics building. It sends it sends positrons around the ring, they're wiggled up and down, and they produce lots of X-rays, which we use. This shows a close-up of where the sample went. So you can see the sample sits on a crystallography support right here, which is on a rotation stage. The X-rays come out of this little hole here and hit the sample. To keep the sample cold, we have this is a nitrogen gas cryostream. So it sends a 100 Kelvin nitrogen gas stream at the sample, keeping it cold. And then when we're ready to warm the sample, we have this little narrow slot. It acts as an air blade. We turn on high pressure gas, and it blows across and shuts off the cold gas flow, or blocks the cold gas flow. And using the same gas source, the sample is then blown on using room temperature gas from this direction. And we've directly measured what goes on here with thermocouples, and it takes about 20 milliseconds to switch from the cryostream flow to warming. So here's at top there is a bovine oocyte that we screwed up. We accidentally thawed it and then slowly refroze it. So it's basically a snowball. Looks like a snowball. And bottom is the X-ray diffraction pattern. And what you see is a lot of rings. Each one of those rings is at an angle that corresponds to a particular lattice spacing in hexagonal ice. So d sine theta is n lambda is the Bragg equation, and so the different d spacings, lattice plane spacings in ice give you rings at different angles. So you can see, so those rings that do correspond to all the rings expected for hexagonal ice. And you can see that they're quite lumpy. They're not smooth, and that tells you that there are ice grains of significant size, large numbers of not too tiny ice grains within the sample. What about if you don't screw up, if you actually just look at cold samples that have been frozen, either using the manual standard approach or using the fast approach? Depending on the cryoprotectant concentration, you can see all of the patterns shown here. The first three on the left, A through C, if you look, the oocytes look clear. They have a sort of opalescence because of the fat content and other stuff inside, but they're clear, as clear as they get anyway. And you can see the only diffraction you see is from water. Water produces two very diffuse rings at different angles. D through F, we have increasing cloudiness and increasing amounts of ice. In D, that diffraction pattern corresponds roughly to cubic ice. You're only seeing ice diffraction at angles that are roughly what you'd expect for cubic ice. And then as you go down, you start to see some additional peaks or additional rings at other angles corresponding to hexagonal ice. And then the last one, you're seeing all the peaks corresponding to hexagonal ice. You can look at those patterns and integrate them azimuthally, so integrate them around the circle. So you then get a plot like this, where each peak tells you the total intensity in each ring. And so that's what's shown for samples A through F here. And if you look at look at D and E, they're kind of funny looking curves, right? They're sort of bumpy, and so let's think about what that might mean. So up top here, this is the expected diffraction pattern for hexagonal ice. And this is the expected diffraction pattern for cubic ice. It turns out none of these exactly match either cubic or hexagonal, and that's a general feature. What they do match is something called stacking disordered ice, which is, it's basically, you can imagine you start making a cubic crystal, stacking plane after plane, and then you make a mistake and you put in a hexagonal plane instead of a cubic plane. So that's what stacking disordered ice is. And so actually what happens as you go from go along the sequence is that the hexagonal fraction goes up and the cubic fraction goes down. This is well known from lots of other studies of both pure water and of aqueous solutions. So what happens during warming? So you may start with a sample that either has no ice after cooling, or it may have some ice. And then what happens? So here I'm showing the diffraction pattern in the case where there's a very weak and diffuse cubic ice diffraction after cooling. So you see you see these rings here. They aren't as wide and diffuse as for pure water. They're coming from very small grain cubic-like ice. So once you start warming, so initially nothing much happens, and then you get to a temperature and where all of a sudden the ice rings, these fuzzy rings, collapse into sharp rings, and then you start to see new rings appear. And then eventually those rings fade out and disappear as the sample fully thaws. So one thing to note is that the total amount of ice is a lot bigger during warming. You get a lot more ice during warming than you had during cooling. The second thing that's interesting is that even though you have lots of ice, during this fast warming, the grain size remains very small, too small for us to make any kind of reasonable estimate of it. It's probably below 200 nanometers in size. So these rings remain nice and smooth. They don't get lumpy at all. And that's a very general feature of cooling and warming, even with the standard protocols as we've implemented them. So one problem in a lot of studies of cryopreservation is, well, how do we measure the temperature of the sample? And how do we measure the cooling and warming rates? We can use thermocouples, but how accurately are they actually recording what's going on inside our cell? And what's really cool is that, well, as long as you've got some ice in your sample, that ice will have a certain unit cell size, and that unit cell size depends on temperature. So we can use the tools of protein crystallography to figure out what the unit cell dimensions are as the ice and the sample warm up, and then convert those unit cell dimensions into temperature. So we have a direct readout of the temperature of the embryo or oocyte or whatever sample we have, without anything touching it to mess things up. And that's what's shown here, just showing the unit cell volume during warming of 3 different samples. And so one thing we learned from this is that the diffraction patterns from the ice structure doesn't change much until you get up to about 180 or 200 K. That's because we're warming reasonably fast here. If you warmed slowly over a day, that wouldn't be true, right? They'd start changing as soon as you got above 100 and above the glass transition, certainly by the time you're above 150 K. But we're warming so fast that really there's not much evolution of the ice at all until you're up above 180 K. The other thing we can directly measure is that cryotops give you 3 to 5 times smaller cooling rates than the crystallography supports. Okay, so let's now look. Here's a case where we use 50% of the standard vitrification solution in our soak, bovine oocyte, and we use the fast cooling protocol. This sample showed no detectable ice after cooling, but you can see that during warming, it does develop ice. In fact, the ice rings actually get pretty strong, even though it starts off with no ice. Okay, and then eventually that ice disappears. On the other hand, if we use 100% strength cryoprotectant solution, 100% strength, that's shown down here, it also starts out with no ice, but it never develops ice during warming. None, 0. Okay, so what this shows, as far as we know for the first time experimentally, or at least certainly as robustly, is that we can cool and warm at least bovine oocytes without forming any ice, using standard cryoprotectant concentrations. So you can repeat these measurements looking at the intensity of the ice diffraction that forms during warming. It turns out the intensity of that ice tells you roughly how much ice, what the volume fraction of ice is in the sample. So you can repeat that for different cryoprotectant concentrations. And what we find is that generally, if you see ice forming during warming, all of the water, all of the free water in the cell is crystallizing. It's not like you're just getting a few percent. And that's mainly because you go from having a few percent to having the whole thing in a very narrow range of cooling rates or CPA concentration. So it's just hard to hit a sweet spot where it doesn't all go. And so this is showing you, this is the ice diffraction intensity as a function of CPA concentration during warming. Okay, so this is the maximum intensity of ice. And it goes up as you reduce the CPA concentration in roughly the right way to be consistent with the free water fraction in the cells. Okay, to summarize these results, and I'm almost done, these are from our studies we did in 2022 and 2023 on oocytes. We don't see any ice diffraction after cooling in standard cooled samples that have 90 and 100% of the standard CPA concentration. If we fast cool, we see no ice after cooling all the way down to 50%, half the full strength concentration. During warming, all of the standard cool oocytes develop strong ice diffraction, consistent with all of their free internal water crystallizing, even at 100% CPA concentration, even though no ice forms during cooling. So this is consistent with the observation and the idea that most of the damage that matters happens during warming. With the fast cool samples, we saw no ice during warming at 100% CPA. So then we've gone back and done some more experiments last fall. This time we're using bovine embryos. They're about 50% larger in diameter than oocytes, so you'd expect them to cool and warm more slowly and therefore be harder to cryopreserve. On the other hand, we've improved all of our protocols since those experiments. There's a different person doing the sample prep. That helps a lot. There were some deficiencies in how the CPA soaks were done. They weren't done in a way that should have in the standard way last time. So that's been improved. We've improved how we handle the samples before cooling and also improved how we handle them after cooling. We had big problems with frost forming on samples after cooling, which then meant that we couldn't make sense of a lot of the data from many of the samples. Okay, so with these larger, slower cooling samples, what do we find? So I can show you, this is a little movie. So this is a standard cool sample, okay, with full strength CPA soak during warming. And so it's on a cryotop. That's why there's other strong rings there. I'll play this again for you. So those rings you see initially, that's just from the plastic of the cryotop. And then you'll see there as you warm, ice diffraction forms, it becomes cubic and then disappears. Okay, so that was true for all the standard cool ones. Once again, they always form lots of ice during warming. So what about fast cool? So here's a case where there's no ice after warming, 100% CPA. And it looks like absolutely nothing happens in this case. Let's see that again. Okay. Actually, you can see the water ring move in, but that's it. Okay. So what we see now is in the fast cool group is that we see no ice form during warming, down to 80% CPA, and even at 60%, it's pretty weak. So we're kind of borderline there. And we have to go all the way down to 50% before we see ice. Okay, so I've got that there. And so we've shown we can cryopreserve bovine oocytes and embryos without ice formation. And even in the case of bovine oocytes, we can even knock down the CPA concentrations a bit and still do that. Turns out, though, we can do much better than we've done already. As I said, here we're warming with just a nitrogen gas stream. And the warming rates we measure on crystallography supports are about 150,000 degrees C per minute. That's better, or at least as good as the largest warming rates that have been reported in the literature. But if you're warming in a thawing solution, you can do a lot better. You can get up to well over 1,000,000 degrees C per minute. And with that kind of warming rate, it seems likely that we should be able to get ice-free cryopreservation at roughly half of our current cryoprotectant concentrations, at least of the penetrating ones. So my final slide. So with the right sample holders and optimized instruments for convective cooling and warming, it looks like you should be able to do ice-free cryopreservation of anything that you can get adequate amounts of CPAs into that's small enough. And what that means is that we've long assumed that ice formation is the major cause of damage in cryopreservation. Well, now we can see if there's anything else that's going on besides ice-related damage in the cooling and thawing cycle, and hopefully develop methods to address that. So how does all this affect biological and reproductive outcomes? We have quite a bit of results already, but more work is needed. So I'll stop there, and thank you for your attention. What time is it, by the way? I didn't set a clock. So
5:22. Perfect.
Okay,
Perfect.
good. Not too over.
No, perfect. Thank you, Robert. Are there any questions? If not, why don't I start? The volume you put on that cryo loop is very important. And
Yes.
I know in the practice in the field, IVF field, that is an issue because a technician or someone is trying to do it uniformly. But even if you get slight differences, the cooling and warming rates will be very different. How do you control that, or
Well, so with what's nice about those crystallography supports is that you just put it on there, and there's a limit to how much liquid can be held on, right? So there's enough, and you don't have to do much. And even if you have some extra liquid, if you double the volume by a factor of 2, that's not a huge deal in terms of outcomes, I think. But when you're using a flat support like a cryotop that's so much bigger, it's a lot easier to get more. There's more scope for variability in how much liquid's transferred, and that could be a factor in why the reproductive outcomes are variable. Is that how much liquid's there? Because of course that affects both cooling and warming.
Right. But you're saying the sweet spot is relatively, the tolerances are pretty good.
Yeah, at least, yeah, I think so.
And how do you eliminate the nitrogen gas above the liquid?
Yeah, so our first paper on this back in 2006, my grad student, well, his initial experiments where he just literally blew it away with his breath. And then he got, a line with compressed nitrogen, a nozzle, and blew it across the top. And actually, you don't do too badly doing that kind of simple stuff. But Nanook reduces the thickness of the cold glass to less than 100 microns. Meaning if you drop a thermocouple down to the liquid nitrogen surface, it doesn't really start to drop until you get within 100 microns of the surface. And that's probably not essential when you're talking about something that's 100 or 200 microns in size, but it was critical in that cryo-electron microscopy vitrification of pure water to do that. But it does matter, these things matter. I think the biggest thing is that not only is it fast using the instrument, but it's highly reproducible. And it's hard to get that reproducibility, even if you try to blow away the cold gas.
Yeah. And we have several questions in the chat. So Kurt Young's asking, thank you for an informative presentation. What suggestions do you have to try to replicate ultra-rapid cooling of livestock embryos under field conditions where use of your machine likely would not be feasible?
Well, I would love talk with him in detail about this. If there's some way I can get your information, that'd be great. Because we'd like to, we're actually, so at my company, we're trying to develop technology specifically for this field. And we have certainly talked about a portable instrument, but what, you know, what would be acceptable? We just watched this Russian documentary called Gunda about pigs on a farm. And so I have some idea of what your conditions are like, and I'd like to learn more about how to make it really work. Well, what's really required to make a useful solution for you? And I think probably everyone on this, everyone here would like to know that too, right? Well, what would really be required? So now that we know that this can work in principle, you know, how would this look in the field?
Yeah. So Kurt, feel free to reach out to Rob, and you have open invitation there. Harry Yu, I would like to ask if you have explored applying this cooling warming method and X-ray ice detection method to multi-cell systems like monolayers or spheroids.
We have not, but we'd love to. And basically, you know, the X-ray experiments are really extremely trivial for us at this point. All we need is to be provided the samples. And if people who are within driving distance or flying distance, or if they can be shipped, it's, you know, we could get a quick publication out. You know, it's all of the data we did most recently was done in 1 8-hour shift at the X-ray source. You could just do so much that, and the X-rays are just great because how else are you going to tell this stuff, right?
Yeah.
It's just the perfect probe for this. So, and it's really fast and easy. I don't want to understate just how easy, once you've got everything set up, these measurements are. We could do high throughput screening. We just need people to send the samples.
That's remarkable because I
So
don't.
that's it. That's an invitation. I mean, and the reason why it's easy is because we're using the entire infrastructure that's already been developed for protein crystallography. So.
Yeah, those were impressive. One more question here, Mark Woodward. What is the explanation for the exponential relationship between CPA concentration and CCR/CWR?
So it's given, it was actually, I had a spectacular graduate student who, with one of my theory colleagues, figured out the explanation. And it's extremely simple. So you start with classical nucleation theory. You have some critical nucleus, and you know, you minimize the free energy, and you come up with some critical radius at which the free energy barrier goes to 0. So that's classical nucleation theory. So what changes when you have solutes, cryoprotectants around? Well, now your critical nucleus still has to be pure water. The critical nucleus still has to be pure water. But you've got all of these cryoprotectant, all these solute molecules floating around. So the probability of your forming a cluster of the critical size that is just water and doesn't include any cryoprotectants, that there's a distribution for that. It's a Poisson distribution. And it turns out that directly gives you the exponential dependence that's observed experimentally. So it's basically, the key idea is that to form the critical nucleus, you have to form a fluctuation of pure water of the size of the critical nucleus that doesn't have any of the other solute molecules present in that volume. And a little stat mech then just gives you that exponential relation.
There are a few more questions, but we're about time. What would you say is the possible reasons for ice diffraction at 180 Kelvin to 200 Kelvin?
Well, so what's happening, you know, as you warm, first, as I said, you're always getting tons of nuclei, no matter how fast you cool. in the cold sample. And when you warm, once you get above the glass transition temperature, the water molecules start to have some mobility, meaning they can wander around and start sticking on to either forming new crystals, new nuclei, or attaching themselves to existing ice nuclei. Turns out that that mobility remains pretty small until you get up to around 180 or 200 K. And there's actually studies back in the early 2000s looking at ice formation in protein crystals. And basically nothing much happened until they got to about 180 K or so. So these are consistent with that. And it's just that as you warm, the mobility of the solvent increases until eventually, the growth rates really start to pick up, and you start forming big enough ice crystals to form diffraction.
Let's take one more question if you have time, Rob. It's the cooling samples in liquid propane instead of liquid nitrogen, about 7-fold difference in cooling rate. And the question is why not to use liquid propane as opposed to liquid nitrogen? It doesn't suffer from Leidenfrost effect.
Yeah, so absolutely. The reasons not to, I mean, that's what cryo-EM does. But they would really love not to have, they use ethane actually. They'd really like to not to have to use ethane because it's flammable. You got to condense it for each experiment. Their health and safety officers aren't wild about having grad students working around it. But that said, if you really want to push it to the limit, cooling rates to the limit, you're going to want to use ethane or propane. That said, so we've done some, it turns out maybe whoever asked the question knows some better data from the engineering literature, because literature I've been able to find, the measurements of cooling rates in different liquid cryogens, they aren't great in that maybe they're using some giant thermocouple, or they aren't using a consistent thermocouple size, or they just don't look like great measurements. So we tried to do some good measurements. What we found is boiling nitrogen versus ethane's about a factor of 11 in cooling rate. If we pump on the nitrogen to cool it to just above its melting temperature, that drops to a factor of 4. Pumped nitrogen is way easier to deal with than ethane, and that may be a good compromise. But we'll see. There's so much scope to improve the warming rates as well. Just the way the warming is done by hand now is really highly non-optimal. So, from what we see here, it looks like with the cooling rates we can achieve, and with these somewhat high but not as high as they could be warming rates, that those are going to be good enough. But that remains to be verified. And something I haven't talked about is, and of course John certainly knows, and others here know, is this cracking issue. As you keep trying to cool faster or warm faster, you do have to worry about internal thermal gradients and fracturing. I don't have any good diagnostics of that for such tiny samples as oocytes and embryos. That is something I'd love to talk to people about if they know a good diagnostic for that, because we can see all the ice now, but there are other things that could go wrong in the freeze-thaw cycle, and what good diagnostics do we have for those? So that's an important open question. How do you diagnose microfracturing and cracking in membranes and things without doing cryo-EM sectioning of your sample or something like that?
I would say sound.
Think so?
Well, we have to talk about it offline, but I think sound would be a great first way to do it.
Okay.
Well, we are a little bit over time, but thank you, Rob. This was a wonderful presentation, and anyone who would like, there are a few more questions, you can email them to Rob, and I'm sure he'll get back to you.
And
Thank you.
I hope this is the beginning of a lot of interactions
We
with
hope so
your
too.
center.
And thank you for accepting our invitation, and very nice to see you.
Yes.
Thanks very much,
Yes.
Rob.
Thank you. Bye.
Bye-bye.